So our population is aging fairly rapidly. So in 2015, about 19% of the US population was 65 years or older. In 2050, it's projected to be 27% of the population. Okay, so we're all, the world is getting much older. So in the US, we're living longer. Our average life expectancy for women is about 81 years. For men, a little bit less, 76 years. So this is great, we're living longer, and that also means that that's more time for damage to accumulate. So when we think of sort of aging, we can kind of think of it essentially as accumulation of damage. Now, when we look at the leading causes of death in those 65 and older, the top 5 are heart disease, cancer, chronic respiratory disease, stroke, and Alzheimer's and related dementias. Now, what's remarkable is that in the last 15 years, death from heart disease has decreased by 11%. In the last 25 years, death from cancer has decreased by 25%. And since 2000, so 19 years, death from Alzheimer's has increased by 145%. Okay, pretty significant. So a few things about dementia. It's the 5th leading cause of death in those over 65, which we just showed. That amounts to 5.7 million Americans, or about 1 in 10 people, that are currently living with Alzheimer's. And by 2050, it's projected to be 14 million. Okay? Now, there's no cure as of now. And this is really a major concern for older adults, and especially women. Women tend to be affected more than men at about a rate of 2 to 1. So you might think, okay, well, if you live long enough, isn't cognitive decline just sort of inevitable, right? Doesn't that just kind of happen? And we can start to glean some answers to that by looking at these regions called the blue zones. How many of you have heard of the blue zones? A lot of you. Awesome. Okay, so these are regions across the world where people commonly live past 100 years of age. And they're not just living to 100 years of age, they're living to 100 years or more, and they have excellent health. Okay, so the places are Loma Linda, California, Nicoya, Costa Rica, Sardinia, Italy, Ikaria, Greece, and Okinawa, Japan. So researchers have gone to these places to try to understand a bit about what's going on. Why are people there living to such a ripe age? And what they're finding about these places is that 1/3 of the population in these 5 regions live to age 90. Right? That is considerably more than here in the US. In fact, 2.5 times more than here in the US. They have 20% less cancer, 50% less heart disease, and virtually no dementia. Even at 100 years of age, these people don't have dementia. So here's this picture of Ikaria, and it's been dubbed the island where people forget to die. Because it is not unusual for there to be, 105-year-olds, 110-year-olds here. So really remarkable. So that brings us to my study. So my study is about women's healthy brain aging, and it's really a unique partnership. So the study started in 2010, and from a few different sort of angles. So part of us, so I'm at the Minneapolis VA, I work in the Brain Sciences Center there, and this gentleman at the top, Apostolos Georgopoulos, he actually did a talk here several years back. Some of you might have been here for that. He is the director of our center, and his wife, who's pictured here on the center picture, she was the director at the time of the Women Veterans Comprehensive Health Center at the VA. And she was noticing that she was having women come into her clinic, women veterans, in their 90s, in the middle of winter, still driving, very sharp. And she just noted, this is remarkable. We need to do something about this. We need to study this. So they started the study in 2010. With the help of a group of women here in the Twin Cities. This is Anita Kunin, Sally Kling, and Barbara Forster. They're women here, local women, who recognize the importance of studying brain health, and they essentially raised money for us to begin this research. Excuse me. And then the University of Minnesota, we're all affiliated there. They were also involved in this partnership. Now, the overall goal of our research is to provide the world's first comprehensive, multimodal, integrative assessments of brain status as it relates to cognition, genetics, and lifestyle across the lifespan. That's kind of a mouthful. I'll unpack that here in the next couple slides. But essentially, the guiding questions of our work is how does the brain change over the course of the lifespan to promote healthy functioning, and why are some brains more resilient than others? So I have a picture of Betty White here. Betty White's 96. And she's kind of like, when I think of healthy brain aging in women, I think of her, right? Because she's still, up until a couple of years ago, was still on TV, still functioning quite well. I haven't seen her recently. Hopefully she's still doing well. But this is sort of what, we all would like to be like that in our 90s, right? We want to live long, healthily, not frail, right? Have any of you heard of, Harmonica Pete? Anyone heard of him? So he's another person who comes to mind when I think of this. He's a 96-year-old World War II veteran who is known for playing the Star-Spangled Banner at various events. Yeah, you saw him. He was recently on the news because he did a big opening at, I think, a Knicks game or something like that. And he opened a Vikings game that I was at a few months ago. So 96, and he's out there playing his harmonica. So remarkable, right? So that's what we're aiming for here. Now, the aims of our study are to characterize brain status with comprehensive brain imaging, and we do a cognitive evaluation. So we have this longitudinal study, this one that started in 2010. We have about 300 people that participate in the study, and they come in every year. And we do some cognitive evaluations, and we do various types of brain imaging so we can get a really clear picture of the brain, its status, from various different parameters, basically. And then we're interested in associating the information we get with regard to the brain with things like genetic information and lifestyle factors like diet, exercise, social connection, that kind of thing. Now, in addition, we want to study the brain changes over time, right? So these people come in every single year, so we can start accumulating vast amounts of data on these individuals, with the ultimate goal being able to forecast who is going to go on to develop cognitive problems, do we think? So can we predict that? And if we can we ultimately intervene? So that's where we are now. So this figure down at the bottom, essentially what this says is that as we get older, from birth into about middle age, our functions or abilities, our cognitive functions, tend to increase and they kind of reach a peak, right? And then at that point, there's 3 different trajectories that people typically follow. So the longer sort of dotted line is what we think of as like normal aging, right? Below that is cognitive aging. So this is shifted over a little bit to the left in terms of time. So this is individuals that earlier than this normal aging are going to start showing some cognitive problems. But then the solid line, the top line, is what's referred to as successful aging, right? So these are people who might have some cognitive problems, but they're going to start much later in life, and they never kind of dip nearly as low as these other trajectories do. So ideally, we'd like to be able to bump everybody up to that successful aging trajectory. Now, when we think of aging and people, you can kind of think of them the same way you would like an antique car, your grandmother's china, that kind of stuff. And that there's 3 main themes or factors that are involved. The first is what I'm calling infrastructure. And essentially what I mean is that some people just aren't built to last as long as other people. And this is really driven by genetics. So I'm going to talk quite a bit about that. Furthermore, we, just like a car, just like China, are going to last longer if we take good care of ourselves. And that's this maintenance piece. And their lifestyle is a big factor, so I'll talk about that a bit too. And then the final part is restoration. So once damage starts to accumulate, can we then restore the brain, just like we might an old car, right? Or fix our grandmother's china. So I'll touch on that toward the end. So the first part is genetics. When we talk about Alzheimer's disease, there's one gene that has been very robustly associated with Alzheimer's, and it's called apolipoprotein E. Has anyone done 23andMe? So a few of you. So when you do 23andMe, you can choose whether or not to find out if you have this particular gene. That's how robust this association is. and with some caveats that I'll get to later. Excuse me. So this is the strongest genetic risk factor for Alzheimer's, and it works in this dose-dependent way. So you can have 0 copies of this gene, you could have 1, or you could have 2. If you have 0, you're what's considered a non-carrier. And in non-carriers, there's still about a 20% chance of people developing Alzheimer's disease. And if they do, they tend to be fairly old, about 84 years old when it develops. If you have 1 copy of this gene, that's called heterozygous. With 1 copy, you have a 47% chance of developing Alzheimer's. And the age of onset's a bit younger, about 76. And if you have 2 copies, there's a 91% chance of developing Alzheimer's. And the age of onset is in your 60s, 68. Fortunately, this last column, this homozygous column, is very rare. Like less than 2% of the population has this, okay? So what this tells us is that having this gene doesn't mean that you're going to develop Alzheimer's disease. And as we see here by the non-carriers, you can have Alzheimer's disease without this gene. So this gene can't be the sole determining factor. And for a long time in the field, in terms of genetics, it seemed like it was. That's the perception that people had. That doesn't seem to be the case. Now, if people have this gene, it's been studied for decades now, and we know that it is associated with a lot of gain of toxic functions and loss of very important functions. So I have a couple of them circled here because I'm going to talk about them in a few minutes with relation to some of our work. In particular, people that have this gene tend to have aberrant brain activity and tend to have increased atrophy or shrinking of the brain. Okay? So keep that in mind because I'm going to come back to it. Now, a lot of our work the last couple of years is focused on a different gene. This gene is called the human leukocyte antigen gene. And these are genes that code for proteins that are on the surface of your cells that are involved with your immune system. So the goal for these genes is to detect and eliminate any foreign antigens. So any viruses, bacteria, anything, these genes come into play, okay? Now for that system to work effectively, it requires that the genes that you have, and you only have a handful of them, but that your HLA genes match with epitopes or little fragments of the virus or bacteria that's floating around in your system. Without a match, these antigens seem to persist and cause damage. They can cause sort of chronic low-grade inflammation, brain atrophy, that kind of thing. Now this particular region of the genome is very highly polymorphic, the most highly polymorphic in the entire genome. And that being said, there's still some genes that are more common than others. So one that I'm going to focus on here is called DRB11302. This gene is, again, one of the common ones, and it's well known to be highly protective against multiple conditions. So numerous autoimmune disorders, MS, rheumatoid arthritis, psoriatic arthritis, Sjogren's disease, things like that. If you have this gene, you don't tend to develop any of those. And research in our lab has also shown that it's highly protective against what we call neuroimmune conditions. I'm going to tell you a little bit more about one of those here. So, excuse me. So as I mentioned, or I think I mentioned, I'm actually housed at the VA Medical Center. So a lot of our work is with veterans. And there's a condition that affects about a third of veterans that were deployed to the 1990-1991 Persian Gulf War, referred to as Gulf War illness. So this condition involves chronic diffuse symptoms, and the etiology has never been clear. This has been studied now for 20-something years, and we still don't have a great idea exactly what this is caused by. But it causes all sorts of cognitive difficulties, lots of memory impairment, things like fatigue, joint and muscle pain, respiratory problems. So it really tends to affect the whole system. And for many years, doctors were working up these veterans, and they weren't finding any evidence of really anything wrong. So for a long time, it was thought to be psychological, and these veterans were sort of just dismissed. And we have come to a very different conclusion. I think the field in general has. But here's some work that we've done with Gulf War illness, where the image here with the yellow arrow, this is the back of the brain, the cerebellum, in a Gulf War veteran. And the other image is an age-matched control. So somebody that's healthy, of the same age. And you can see a pretty significant difference in that part of the brain. Okay, so we're showing that there are very clear brain damage effects that are associated with this condition. Now, we wanted to then talk about or look at how that was related to this human leukocyte antigen gene that I just said was protective, right? This DRB11302. And what we found is that those individuals deployed to the Persian Gulf War who had this gene, their brain volume was significantly greater than people that did not have this gene. So there's something about this gene that was protecting them from having brain atrophy. Okay? So this, even though it's totally unrelated to aging, started us thinking, well, wait a second, aging is associated with brain atrophy. Starting at about the age of 40, our brain tends to shrink or atrophy at the rate of about 4% per decade. So we were wondering, okay, could this gene have any protective effects in what we see with aging? So that led us to this particular study, where we did essentially very similar to what I just showed you, that we looked at volume, right? How much brain. And what we found is that in our study of our participants, this longitudinal study, that if you do not have this gene, that the individuals did tend to show the expected pattern of atrophy over the course of the lifespan. So it ended up being about 3.8% per decade. But the people that have this gene, there was zero atrophy, absolutely no change. Okay, so even here, with the aging, this is protective. Now, interestingly, the ApoE gene, we looked at that here, it had no effect. Okay? So this isn't driven by this ApoE that's been associated with dementia before. So that was great. So then we wanted to look at, what about brain function? How is this working in terms of brain function? So we had mentioned that ApoE4 is associated with the aberrant brain activity. And what we found was similar and even better, that people that did not have this protective HLA gene, and they had the E4 allele, that ApoE that is associated with dementia, had significant deterioration in their brain function over time. But people that had the protective allele, even if they had E4, their brains got better, right? They actually got more efficient over time. So again, ApoE4 isn't the end-all be-all here. There's something very significant going on with this human leukocyte antigen gene that's involved in the immune system. So after we wrote that paper, somebody published a commentary where they said, this particular gene, there's a future here, kind of shaking up the field a little bit. And we couldn't agree more. We think this is shaking things up quite a bit. So since then, we've taken this to go from looking at individuals here in our study, local people here in Minnesota, to looking at the population. And then for this particular study, we looked at continental Western Europe. This part of the world has the highest rates of dementia. So, excuse me, we were interested in looking at the association between dementia and this particular gene in 14 countries in continental Western Europe. And we find essentially the same thing, that as the prevalence or the frequency of this particular gene increases, the prevalence of dementia significantly decreases. This is a highly significant exponential fit here. And what that translates to is that as the frequency of people carrying this gene doubled or increases by 100%, the prevalence of dementia is decreasing by 1/3. Okay? So pretty remarkable. So this is really a breakthrough in aging research. So it's showing that this gene that's related to immunity protects against age-related changes in brain structure and function, and it protects against dementia. By the way, all of that data I just showed about Europe, we also accounted for ApoE, and it didn't have any, it didn't change things. So even with these at-risk genes, these protective immune-related genes are pretty significant. And this is really just the beginning. So now we can start looking at things like, well, what are the mechanisms here? Can we intervene? What about other protective HLA genes? Remember I said this is the most highly polymorphic, meaning that there are tons of different types of alleles that are out there. So we're starting to answer all of these other questions. And to do that, we can kind of work backwards in order to move forward. So remember that the goal of these genes is to eliminate foreign antigens, so little bits of viruses and bacteria. So then we can then look at, well, what antigens match with this particular gene, okay? And what we know, this is just a little sample of some of them, but things like the HPV virus, various herpes viruses, which most adults have herpes virus or have been exposed to herpes virus of some sort throughout their lifetime, but also influenza, right? So things that are very common, this particular gene is a match for, which means it helps the body eradicate any of things that might linger around, okay? Now herpes viruses I do have highlighted here because they have been, there's prominent theories suggesting that herpes may actually be the cause of dementia. So that's something that we are kind of looking into as well. So if you have this gene, it wouldn't matter, according to this. So what we've proposed is that if you're exposed to a pathogen or some sort of harmful persistent antigen, if you can eliminate it by your HLA, particularly this one we know is so highly protective, DRB13, you're fine, right? If you can't, if your body's not able to eliminate it, that then produces some neuronal damage over time, right? This is going to be sort of like a slow kind of chipping away at the brain. And we think that ApoE then becomes an important factor. It's kind of a secondary downstream factor. I had mentioned ApoE4. What I didn't mention is that there's other types of ApoE as well, ApoE2 and ApoE3. And these particular types, the 2 tends to be highly protective against dementia, and ApoE3 tends to be neutral. So what happens then when the neurons start to be damaged, the ApoE gets activated. And if you have an ApoE2 or 3, that then facilitates neuronal repair. So your brain kind of gets back in shape. If you have the ApoE4, that actually creates more damage to the brain. The ApoE4 is what we call neurotoxic. So it has these little fragments that break off and damage the brain a little bit more. So ApoE is definitely important, and it's not the end-all be-all. It definitely seems to have some secondary effects here after the immune-related components. So I'm guessing that some of you are probably thinking, well, 1, how do I find out if I have this gene? And 2, what if I don't have this gene? So this isn't like commercially available right now. It's not something that people can find out. So I just want to make sure that's clear. But if you don't have it, it's probably okay for most people, as there are lots of other protective genes. We had said this was highly polymorphic, so we already know, we've already identified several others that do seem to be highly protective in terms of brain health across the lifespan. The second thing is that it's really important you protect yourself from pathogens. So this means like hand washing, right? The stuff your mom told you years ago. It's actually really, really critical that you protect yourself against all sorts of viruses and bacteria. And then finally, important that we really take care of our brain. And that brings us to the second point, this maintenance or lifestyle piece. So what do we mean by maintenance? Well, essentially, I just mean, how do we keep the brain healthy across the lifespan? So the brain is this massively interconnected network, hundreds of billions of neurons that are interacting there. And throughout the lifespan, this network is constantly adapting and evolving. And in the right circumstances, the brain should get better with age. And that's kind of what we were showing in that earlier figure when I was talking about brain function and improvement. The brain becomes more efficient. And that's ideal. That's what ideally we'd like to have happen. So I have wine here. It's kind of like a fine glass of wine, right? It gets better with age. So that's in the right circumstances. So what's the problem then? Well, the material that the brain is made up of is perishable, and it is subject to all sorts of insults. And there are lots of repair mechanisms in place. Your brain is constantly trying to repair itself. But over time, the need for repair can outweigh the ability to repair it. And that's when we start seeing significant changes. And that's shown here. You see the Alzheimer's brain is much, much smaller than a normal brain. So that's really when the damage is pretty significant. So healthy brain aging then is essentially looking at how do we preserve the cell biology? How do we avoid disease? So, excuse me. So there have been decades of research aimed at this, and recently in 2017, the Executive Summary on Dementia Prevention, Intervention, and Care came out with a list of factors, potentially modifiable factors, related to Alzheimer's disease. And they looked at across the lifespan. So we know that having less education, particularly in your early life, is associated with greater risk for Alzheimer's disease. For some reason, they skipped from 18 to 46. So I guess that period of our life isn't that important. But once we hit midlife, there's a few factors here that do seem to be highly related to developing dementia. And these are hypertension, obesity, and hearing loss. And then when we look later in life, at 65 and above, the factors that are associated with dementia are smoking, depression, physical inactivity, social isolation, diabetes, and hypertension. Now what's remarkable about this is that they stated in this paper that interventions, like these are all areas that we can intervene on, right? And that interventions might have the potential to delay or prevent 1/3 of dementia cases, right? So there's lots of room for hope here. This is great. Now they took all of those factors and they put it in this fancy Venn diagram, and to sort of try to explain what areas these different factors are working on. So the mechanisms. So there's 3 basic mechanisms. The top there is cognitive reserve. On the left-hand side, we have reduced brain damage, things like vascular, neurotoxic, and oxidative stress. And on the right-hand side, these are the factors involved in reduced brain inflammation. So I'm going to talk a little bit about some of these. The first one is cognitive reserve, up at the top. So having preserved hearing, having continued education or higher education, potentially cognitive training, it's a bit controversial, but also having a rich social network, reduced depression, and exercise all fit into this idea of cognitive reserve. And cognitive reserve is essentially just building up your brain to be as big and strong as possible to allow you to compensate for any damage that occurs. Okay? So this has been emphasized for quite a bit of time now. So coming to things like this, lifelong learning, that was like part of the description of the Humanists in Minnesota, right? That is this, right? That is cognitive reserve. You're learning throughout your lifespan. You're continuing to build your brain, okay? Now I have these pictures, this picture here. I loved this story. This was, I want to say like February or something earlier this year. This was 2 cribbage players, age 108 and 105, that were having a cribbage battle. So they lived at separate nursing homes. They both were avid cribbage players, and they had this huge battle. They took one from his nursing home, brought him to the other, and they played in front of like all these people. So that is cognitive reserve, right? At 108, they're still very actively working their brain, okay? Very, very important. This whole idea of lifelong learning is so, so critical. The other couple things that I want to point to here are diet and exercise. So our Western diet is not very healthy. I think we all know that, right? And we also know that more Mediterranean style diet, so lots of fruits and veggies, fish, occasional wine, these more plant-based diets tend to be much healthier for us. So a lot of those blue zone countries have a very heavy plant-based diet. And it's borne out in the brain too. So this top image here shows somebody who had followed a Mediterranean diet on the left, and it's someone the exact same age on the right, same in terms of their cognitive health, same in terms of their physical health, but the only difference was basically their diet. And the person on the right, there are big ventricles. We don't want that. That's not good. And a smaller hippocampus. This is where learning and memory that's a critical component for that. So, excuse me. So our diet is very much affecting our brain, right? We think of our brain as being kind of, you've probably all heard of this blood-brain barrier, right? Our brain's protected. No, it's not. It's actually really easy to access the blood-brain barrier. Yeah. The keto diet? Yeah, I don't know a lot about that. So it's a high-fat diet, right? Yeah, I haven't seen studies that looked at it specifically in terms of the brain, what's going on with that. I think generally speaking, the Mediterranean style diets tend to be favorable in terms of both heart health, brain health, that kind of thing. And in all fairness, I don't know a lot about research on the keto diet. So diet's very, very important. Also very easily modifiable. The other thing here is exercise. So exercise is in the middle of this whole diagram, right? This is kind of like the brain trifecta. If you're engaging in exercise, you're increasing your brain reserve, you're reducing brain damage, and you're reducing brain inflammation, okay? Exercise is so very important. Now, it doesn't mean that we have to be doing, I think this is yoga. This is very impressive, right? This woman, like I don't think I could do that. And this isn't necessary. And going to the gym isn't necessary, right? Those things are fine, they're great. But this can mean even just 10-minute walks. Just getting out and being active is so very important for us. And we know that whatever kind of affects our heart also affects our brain. Our brain's just another organ, like everything else in our body. Okay, so anything that affects other organs ultimately can affect the brain, both benefits and harm. So what this executive summary team stated was that there is a cumulative and dose-related exposure to these cognitive reserve enhancing factors like physical exercise, intellectual stimulation, or leisure activities over the lifespan was associated with reduced risk of dementia in late life, even among individuals with genetic predisposition to dementia. So, and that means that E4 allele. So even people that have this allele, if you do any of these things, it's going to be tremendously protective. And if you do more than one, it's cumulative, it's dose dependent or dose responsive. So there's lots of different ways we can modify things. One thing I wanted to point out really quickly here is this rich social network piece, right? I'm like looking at this group. This is what we mean, right? Getting together with like-minded people, engaging socially. I mean, social isolation is not good for many reasons. It tends to create depression, which then perpetuates itself. So being around other people and having a rich social network is really, really critical for brain health too. So kudos to you all for being here and being lifelong learners, engaging in some social activities, etc. Okay. Now, if we think about how these lifestyle factors might work with the genes that we've been studying, what we would expect here is that those people that have this DRB13 or other protective immune-related genes and they engage in healthy habits are probably going to have, pretty high cognitive functioning throughout the course of the lifespan. Conversely, people that don't have these protective genes and they also engage in unhealthy lifestyle factors, they're probably going to show the steepest decline in terms of cognitive functioning. And the others are probably somewhere in the middle. So this is something that we can study with the data that we're collecting. The last part I want to talk about here is this idea of restoration or treatment. So we had mentioned earlier that there is no cure for dementia right now. The current treatments are really aimed at just sort of halting or slowing down symptom progression. So the best option we have right now is prevention, and that really means focusing on the lifestyle factors, modifying your diet, increasing social activity, exercising, etc. But there are several novel treatments under investigation. I thought I'd just briefly talk about some of these. So this first one, excuse me, isn't work that we're doing, but I think it's really neat, so I wanted to share it with you. So it's all about removing what are called zombie cells. So the idea is that as we age or encounter things in our life, our cells, sometimes they get sick, and when they get sick, they're supposed to die, right? They're supposed to have mechanisms in place that they die and it's fine. But there's some cells that don't do that they linger, and these are the zombie cells. So they're kind of ill, sick cells that aren't going away like they're supposed to, and then they're actually making other cells nearby, they're making them sick too. So it's kind of like contagion in your body, okay? So this is research that's been going on at Mayo, and these are twin mice, okay? They were born in the same litter. The one on the right had all of their zombie cells removed, okay? So they're nice and plump, they look, healthy, I suppose, as far as mice are concerned. The one on the left, it has this hunch, it has lost a lot of its hair, and this is sort of an aging mouse model. So it seems that this is a promising avenue that researchers at Mayo and some people here at the U are looking into. How can we translate this now into humans? How can we remove zombie cells? Now, we think that these zombie cells are related to these viruses and pathogens that people can't eliminate. So if you don't have the genes, these protective immune-related genes, and there's things floating around in your system, that they're going to affect the cells. And that's what we think might be going on here. Now another thing is, another potential is immunotherapy. So this idea, the way that this whole thing works with these protective genes is that you have a small array, about 12 different HLA gene types in your body, on your cells. And that if you encounter an external antigen, so a bacteria, virus, thing like that, if it matches with the repertoire of HLA proteins that you have, then it starts this whole cascade that then promotes antibody production and facilitates eliminating these things from your system. Now, if you don't have a match, then the whole cascade kind of falls apart. The antigens persist in your body, and that promotes disease. So that would often look like inflammation or atrophy, that kind of thing. So this idea that things are persisting is an opportunity for intervention. So I mentioned this Gulf War illness earlier, and that's really the Gulf War literature or research we've been doing has really informed a lot of our aging research and vice versa. So I'm coming back to that for a moment because it's a bit of proof of principle with this immunotherapy idea. So what we have here at the top is we have serum from Gulf War veterans and from control healthy veterans. So we have spreading is the green. You want spreading, you want your neurons to be growing and connecting with each other. Apoptosis is the bottom, the blue and red. The red is the apoptosis, and you don't want that. That's your cells dying. Okay? So we have the control is the left figure. The Gulf War is the middle one, and there you can see they're all kind of clumped together, right? They're not doing the spreading thing that healthy cells should be doing, and the cells are dying. Okay? Now, when we took the serum from a Gulf War veteran, this is like in a dish, not inside the people, and we added serum from the control veteran, corrected everything. Okay? So that's what's shown here on the right, that it went back to spreading, and there wasn't apoptosis. So there's something in that person's serum that's getting rid of all the stuff that's missing from the Gulf War person. And that seems to be related to this HLA. Okay? Now, similar thing here at the bottom. In the bottom, we had veteran Gulf War serum, and we added IgG. So IgG is pooled antibodies. So this is like common immunotherapy in clinics all over the world, that it's got serum from lots and lots of people. They put it all together, and then they deliver it to people to sort of modify their immune system. And the same thing happened here, that when we added these pooled antibodies, these commercially available antibodies, that it increased the spreading and it decreased the apoptosis. So again, we're able to intervene here and sort of ameliorate what's happening with Gulf War veterans. So, so now the question is, okay, great, does this apply to aging? And that's something that we're looking into. So we are collecting serum from our women, and we're trying to identify, can we identify antigens that might be persisting, and can we get rid of them? Is there a hand?
What is serum?
Serum? It's a part of the blood. Yeah. So we're interested in this idea of personalized precision immunotherapy, right? So can we identify what antigens people might have? Can we eliminate them? Notably here, if these antigens persist, I had said before that they're associated with things like inflammation and atrophy. Those are two of the main mechanisms here that are thought to be associated with dementia. Okay? Excuse me. So that brings us back to these blue zones. So we think, certainly lifestyle factors, right? These plant-based diets, exercise. People in these countries exercise, they're not going to the gym. It's just kind of a way of life, right? They're out, they're walking regularly, they're out tending fields, they're doing everything in their life is all about movement and exercise. Those things are certainly related to maintaining brain health in these areas, but these are also really fairly isolated communities. So we think that part of why these people are staying healthy so long is that they probably have pretty limited exposure to foreign antigens. So viruses and bacteria aren't necessarily coming in from outside, and or they have this genetic protection. They have these genes, this HLA has evolved. That's why it's so highly polymorphic. It evolves to get rid of viruses and bacteria in your local area. So if there's nothing new that's introduced, then they have all the genetic makeup to take care of all the local viruses and bacteria that they might be exposed to. So I just want to point out that the research that we're doing couldn't happen without philanthropy. It's been very, very key to our research. Shockingly, there's very little grant funding for healthy aging. We would think that people would want to support that, right? How do we figure out how to help people maintain brain health? And there just isn't. It's quite limited. So our thought is that if we can understand what promotes brain health, then we can have a tremendous impact on disease. And ideally, prevent dementia, intervene, etc. So the study I had mentioned started in 2010. Those women in the community, they raised about $160,000 to start the study. And then a donor provided $1 million to establish the Kunin Professorship. So I was named as the first holder of that professorship, which guarantees 5 years of funding. And then ultimately, we're hoping that this will be able to raise another $1 million, and that would guarantee that this goes on in perpetuity, that we could continue studying this. This is my team. Dr. Georgopoulos in the center was the one who was here several years ago, so some of you might recognize him. But we are a big multidisciplinary team. I'm a clinical psychologist by training, as Mark mentioned, but we have neurologists, neuroscientists, biochemists, all sorts of different disciplines. And as a group, we're all aimed at understanding brain health and disease and various factors related to it.